Welcome back to Med Lecto. As we are discussing hemostasis, in the previous lecture we have discussed vascular constriction, but now we are going to discuss about platelet plug formation. There are three processes which are involved in the formations of the platelet plug. First one is adhesion, second one is aggregation, and third one is platelet release reaction and amplification. Before we discuss these points, we first look at von Willebrand factor. VWF is basically a glycoprotein which have different units. 2 to 50 dimeric unit means they are present in two subunits. Von Willebrand factor is produced by endothelium or platelet. In endothelium, there is a body which is called Weber pallet body, like this. This is a Weber pallet body. It has von Willebrand factor. In platelets, there is a alpha granule which is present in the platelet, and in alpha granule there are a lot of von Willebrand factor present in it, right? In endothelium there are two ways of secretion. This von Willebrand factor is secreted in two ways. First one, it continuously secrete from the endothelium and release into the blood. And second one, it secrete in stress situation, which or different substances like secretagogues. Secretagogues are basically that situation or factors which influence or secretion of the von Willebrand factor. These can be exercise, in case of exercise, adrenaline, or if infusion of the desmopressin occur, then it can also cause release of the von Willebrand factor. In desmopressin, actually this is a drugs which is used for the treatment of the hemophilia A or von Willebrand factor diseases, right? Here is the structures of the von Willebrand factor having different subunits. They are present in multiple forms, right? When they are secreted from the endothelium, they are in full active form or in multiple form. This is a multiple form, and this is active form, and it convert into different dimeric or smaller subunits with the help of metalloprotease enzyme. This enzyme convert multiple forms of the von Willebrand factor into smaller multiple forms, or you can say dimeric unit or monomeric units, right? Let's move how platelet plug is formed. Before we discuss platelet plug formation, we have to notice that there are glycoproteins which are present in the membrane of the platelet. Glycoprotein Ia, 5 and 9. This is a complex which only attached to the von Willebrand factor, right? And there is an other glycoprotein complex, glycoproteins 2b, 3a. These can attach either with von Willebrand factor or fibrinogen, right? And third one is glycoprotein 1A, 2A or 6. It only attach with the collagen, right? Here you can see a large blood vessels having 3 layers. 3 layers. Innermost layer is the tunica intima, right? Middle layer is tunica media, and the last one is tunica adventitia, right? As we know that tunica consists of 2 layers. First one is endothelium, which is in direct contact with the blood flowing through this large blood vessels, and just below it there is an other membrane which is called endothelial membrane. Subendothelial membrane, right? If injury occur to the blood vessels, as you know that there is a layer of tissues, and when injured occur like this one, then loss of blood occurs, and in that situation, in the subendothelial layer there are a lot of collagens present in it. There are a lot of collagens present in it, like this is blood loss of blood. In subendothelium there are a lot of collagens present in it. As you know that this endothelium also damaged, and lot of, in case of injured or stress situation, lot of Willebrand factor which are present in the vapor palate body of the endothelium release tremendously. Lot of von Willebrand factor release, and their concentration in the blood will also increase. Like this is a von Willebrand factor. Von Willebrand factor, right? See, this is a normal platelets which is flowing in the blood stream. As you know that there are some glycoproteins present on its cell membrane or plasma membrane. This is a complex glycoprotein complex. This is an other which is shown by the blue color, other complex, and this is third one complex which is shown by the green color, right? After the injury occurred, the platelets which are flowing freely in the blood streams detect, and some changes occur in the platelets, like there several pseudopods protrude from the platelets with the help of different contractile protein present in the platelets. These proteins are actin, myosin, thrombocytinin. These proteins which are present in the platelets contract and change the shape of the platelets, right? This platelet having affinity for the attachment of the von Willebrand factor or collagen which is exposed to the membrane, right? I told you that this glycoprotein is just, which is shown by the black color, is just attached with the von Willebrand factor. Like this is a glycoprotein complex. This glycoprotein complex is just form a bond with the von Willebrand factor, right? And the second one, which is shown by the blue color, this complex glycoprotein B, glycoprotein A can attach with the von Willebrand factor and fibrinogen, like you see this complex, right? This complex can attach with von Willebrand factor, right? And the last is the glycoprotein complex, which is shown by the green color, just only attached with the collagen, which is exposed by the subendothelial membrane. This complex, right, individually attach with the collagen, which is exposed by the subendothelium, right? This is a von Willebrand factor which attach to the collagen, right? In normal situation, von Willebrand factor don't attach with the endothelium. It just attach if the collagen is exposed, right? This formation is called adhesion, right? This whole process which I have just told you, that this is a adhesion process which is occurred in the platelet plug formation, because platelets just attach to the membrane, right? Another platelets come. Why? This platelet release lot of chemicals which can be like ATP or thromboxane A2, right? Thromboxane A2. This platelet release ATP or thromboxane A2, which attracts other platelets come toward them, right? Here another platelet having glycoprotein. This glycoprotein attached to the another von Willebrand factor, like this platelet having glycoprotein complex attached to the von Willebrand factor, and this complex can also attach with this von Willebrand factor, right? It means now two platelets are joined together. Similarly, this complex, right, this second complex can attach with von Willebrand factor and fibrinogen. This complex here, very interesting, this smile face which is showing over the board is a protein. What proteins is this face? Is the fibrinogen. Right? This is a fibrinogen. And the glycoprotein 2B and 3A complex now attach with this protein, fibrinogen, right? Or this complex, same complex, can also attach with the von Willebrand factor, right? And you can see this complex also present another platelets. This can also attach with this fibrinogen or this von Willebrand factor, right? Now, this is the final stage at which two platelets can adhere or aggregate themselves. This process is called aggregation. Aggregation. Right? So this is all about the plug formation, right? Here you can see lot of further platelets come and attach to this side. Here I show you. You can see that lot of platelets accumulate and form a plug. The last step in the platelet plug formation is the platelet release reaction and amplification, right? It means the formation of the plug is enhanced by the different factors like primary activation of the plug formation is release of ADP or von Willebrand factor. These are basically released from the platelets. ADP is present in the electron dense granules and the von Willebrand factor is present in the alpha granule. Secondary activation is basically the formation of the thromboxane A2, which attract the other platelets, right, for the formation of the plug, right? How thromboxane is produced and how they influence the strengthen of the platelet plug, right? You can see endothelium cell on the left side and the platelet on the right side, right? In the endothelium, the phospholipid is converted into the arachidonic acid with the help of phospholipase enzyme. This is an enzyme which converts the phospholipid into the arachidonic acid. Arachidonic acid then convert into endoperoxidases with the help of cyclooxygenase. Endoperoxidases are actually prostaglandin G2 or prostaglandin H2. These are the prostaglandin, and endoperoxidases are converted into prostacyclin with the help of prostacyclin synthase, right? This is the formation of the prostacyclin in the endothelium. And in the platelets, which is present in the blood stream, same process occur in the platelet, but little bit different. First of all, phospholipid convert into the arachidonic acid with the help of the phospholipase, and then arachidonic acid convert into the endoperoxidase through the cyclooxygenase. As endoperoxidase are basically prostaglandin G2 or H2, it convert into the thromboxane A2 through the thromboxane synthase. Here, a different compound, right? As ATP convert into the cyclic AMP, adenosine monophosphate, with the help of adenylate cyclase. Thromboxane A2 has negative effect on this enzyme, which is adenylate cyclase. It has negative effect on them, right? It means it inhibits or hindered in the conversion of the ATP into the cyclic AMP, right? It means if cyclic AMP is not present, then it not convert into the AMP, right? And due to the lack of AMP, right, the calcium concentration is increased, right? You can see cyclic AMP is inversely proportional to the concentration of the calcium. If the cyclic AMP is low, then the concentration of the calcium is high. It means, and the calcium concentration is important for the strengthening of the plug, right? This is due to the negative effect of the thromboxane A2 on the adenylate cyclase. See, cyclic AMP not produced, it means its concentration low and the calcium level is high, right? Another compound which is produced in the endothelium, it has a positive effect on this enzyme. This enzyme is very much important, adenylate cyclase. Prostacyclin has a positive effect on this enzyme, adenylate cyclase. It means it enhances the conversion of the ATP into cyclic AMP and then AMP. It means a concentration of the cyclic AMP increase. It decreases the calcium concentration. It means the plug is not as much strength or as much strong as it should be. So this is all about the platelet plug formation. In the coming videos, we will discuss next blood coagulation and so on. Thank you so much.